attending, joining us today on our second session of the Winter Lunch and Learn series, brought to you by the External Committee at the Graduate College. My name is William, and I will be your moderator for this afternoon session. We're very excited for two of our scholars to have the opportunity to share their research with you today. So we'll start off with some opening remarks, and then I will introduce our presenters. They will each have 20 minutes to share their presentation, and at the end, there will be a question and answer period. So please use the Q&A box to enter your questions at any time during the presentations. You don't have to wait until the very end to ask. And finally, we will conclude our session with a short summary. So I'd like to take this opportunity to acknowledge the traditional territories of the people of the Treaty 7 region in southern Alberta, which includes the Blackfoot Confederacy, comprising the Siksika, Piikani, and Kainai First Nations, as well as the Tsuut'ina First Nation and the Stoney Nakoda, including the Chiniki, Bearspaw, and Wesley First Nations. The city of Calgary is also home to the Métis Nation of Alberta Region 3. I would also like to note that although we are connecting virtually today, the University of Calgary is situated on land adjacent to where the Bow River meets the Elbow River, and that the traditional Blackfoot name for this place is Mohkinstsis, which we now call the city of Calgary. Oh. So what is the Lunch and Learn series all about? Well, first, it's a public presentation series that aims to bridge the Pediatric College with the Calgary community. Our series has been ongoing since April, and we are continuing the series into the fall and winter. During each Lunch and Learn period, graduate scholars present their diverse research for the public. So without further ado, today we have Shiama, who is a master's student in the Department of Biochemistry and Molecular Biology at the University of Calgary. Her research is focused on developing a method of culturing spermatogonial stem cells, which have the potential to preserve male fertility. Her presentation today will be on establishing a co-culture system for porcine undifferentiated spermatogonia. Thereafter, we have Sumedh, who is a PhD candidate in the Department of Community Health Sciences at the University of Calgary, and will share with us a presentation on KidsPRO, which is a tool to support patient and family-centered care in pediatrics. Sumedh's research focuses on how he incorporated novel approaches such as patient engagement and the use of video to recruit patients to his PhD project. This project aims at breaking silos in health research. All right, Shiama, the floor is yours, so please take it over.
Great, thank you. I'm just going to share my screen. So let's start by talking about overcoming infertility after recovering from childhood cancer. Every year in Canada, approximately 1 in 850 Canadian children are diagnosed with cancer. And that means children aged generally 14 years or younger. So due to advances in technology and in therapy development, survivorship has been increasing over the years. At this point, about 83% of those who were initially diagnosed will survive. And this is really great, and it provides so much hope for patients and for their parents and their families. But it's also really important to recognize that a lot of these therapies can be very toxic, and there are a lot of very long-lasting effects of these therapies, one being infertility. And while this infertility does affect both males and females, the focus of my research and what my lab does could potentially benefit the male survivors. So adult males, specifically men, when they're diagnosed with cancer, they're often told to freeze their sperm prior to receiving their treatments so that even if they are left infertile afterwards, they have a bank of sperm saved up and they can still have biological children later on in their lives, even if that is through some sort of assisted reproductive technology. Younger boys, however, don't have that option. They haven't yet reached puberty, and so they don't have sperm and they don't yet produce sperm. But what they do have is a pool of spermatogonial stem cells. And this is really important because spermatogenesis, or sperm cell development, is spermatogonial stem cell driven, meaning essentially that sperm comes from these cells, or these cells will develop into sperm. So what these young boys can do instead is they can have testicular biopsies taken that contain these cells. And then after receiving their treatments and recovering, they can have these cells reintroduced into their bodies via transplantation. And then when they do eventually reach puberty and have a more mature body that can support sperm development, they will have sperm developing in their bodies, and they can have biological children later on in their lives. So this kind of concept of transplantation has actually been developed in many animal models, but has not yet been developed for humans. But we'll talk a little bit more about that later. So in vivo, or in tissue, spermatogonial stem cells have kind of different fate options, and these options are influenced by the spermatogonial stem cell niche, which consists of the cells and factors that surround them in tissue. So here we have a spermatogonial stem cell. And so in terms of fate decisions, it can self-renew in order to maintain that pool of stem cells, or it can differentiate, which will yield spermatozoa or sperm. And then the cells that are in close association and that can influence this decision to self-renew or differentiate include Sertoli cells, peritubular myoid cells, testicular endothelial cells, and Leydig cells. So back to this idea of transplantation and developing this therapy. In order for this to proceed, there needs to be a lot of research and a lot of development. And this research and development requires a large quantity of cells. And unfortunately for us, there are very few spermatogonial stem cells in tissue. So we're starting with very small populations. And in addition to that, it's really difficult to enrich these cells because there's not a marker that is specific to these cells, but not to other cells. And usually that is a thing with other cell types, and this can be taken advantage of in order to separate and enrich cell types. But obviously no such marker exists. So what scientists can currently do is isolate and enrich populations of germ cells and or undifferentiated spermatogonia, because spermatogonial stem cells will be contained within these undifferentiated populations, but they will not make up the entire population. And these two first points about the population size and the enrichment can be really well illustrated in this picture here. So this is a picture I took right after digesting 1 week old porcine testis tissue. So as we can see here, only about 7% of these cells are UCHL1 positive, UCHL1 being a marker for a class of undifferentiated spermatogonia, of which spermatogonial stem cells are present. So this basically means up to 7% are SSCs, but definitely not all of them are. And this also kind of leads into this third point, that transplantation is generally relatively inefficient. And because the populations of cells that are being transplanted aren't necessarily all spermatogonial stem cells, this adds an increased layer of inefficiency to the process. So basically, the kind of bottom line is that we need more cells. We need to be able to generate more of these cells, which we refer to as expansion. So this expansion can be achieved by establishing a culture system. This is a schematic of a very simplified version of a culture system, in which we have our germ cells here, and they will be put onto a culture plate that contains feeder cells and conditioned media. And so feeder cells are basically a cell type that have been treated such that they don't divide anymore, but they do secrete factors that can promote germ cell proliferation and survival. And usually the choice of feeder cell is decided upon due to its ability to secrete factors that mimic the niche. Similarly, with conditioned media, it's regular media or baseline media that has been enriched with factors that support germ cell proliferation or ward off oxidative stress, things like that. And so this whole combo of the dish with the plates goes into an incubator, which provides a controlled environment. And then after a period of time of a scientist's deciding, we have an output, hopefully, of cells in greater numbers than what was the input. So such expansion through culture systems has been achieved in smaller animals. So first with mice, and then the factors that were very successful for mouse expansion, namely fibroblastic feeder layers and media supplementation with the following factors, were applied to similarly expand cells isolated from other small rodents and animals, including rats and hamsters and rabbits. However, similar expansion has not been achieved in larger animals using these factors, and especially with the goal of trying to expand human spermatogonial stem cells for that potential application of transplantation. It's really important to find a translational model and to do research to establish a similar level of expansion for cells from a more translational model. So my lab uses pigs because pigs are physiologically and anatomically quite similar to humans. And this brings us to the purpose of my project, which is to establish a co-culture system that can be used to expand germ cells that have been isolated from pigs. So I'm hypothesizing that undifferentiated spermatogonia, which will include spermatogonial stem cells, will be able to survive and grow in cultures that I have established using either or peritubular myoid cells or testicular endothelial cells as the feeder layers. And I'm hypothesizing this because so far in the literature, Sertoli cells are the one cell type from the niche that have been investigated as a feeder cell for pig germ cell culture. There are very few papers out right now about pig germ cell culture. And of the very few papers, Sertoli cells have been pitted against different types of mouse fibroblastic cells, and the pig Sertoli cells have come out on top. And a reason for this is because they are in the niche, they clearly secrete factors, namely GDNF, that support germ cell proliferation and growth. However, they're also implicated in germ cell differentiation. And it's really important that at the end of a culture, the cells that come out of it are in the least differentiated state possible, because it is only the spermatogonial stem cells that will mature into sperm when transplanted. So I think because Sertoli cells have been the only cell type investigated thus far as feeder cells, it's really important to look at other cell types in the niche. So peritubular myoid cells and testicular endothelial cells are also really great options for feeders because they both also secrete GDNF and other factors that promote germ cell growth and survival. And they haven't been investigated yet, which is why I'm investigating them. So towards my hypothesis, towards investigating my hypothesis, I'll be establishing a co-culture system using PMCs and TECs, the myoid cells and the endothelial cells, as feeder layers for porcine germ cells. And then after establishing these two systems, I will be comparing the survival and the proliferation of the germ cells that grow on these systems with the survival and proliferation of germ cells that are grown on other feeders from the literature, namely Sertoli cells and porcine fetal fibroblasts, or in a no feeder condition. And this is the first time that I am mentioning porcine fetal fibroblasts, but I mention them because previously in studies of smaller animals, mouse embryonic fibroblasts are often used, but a mouse cell doesn't necessarily work very well to culture pig cells. So I'm using an analogous type of cell, a pig fibroblastic cell, and potentially that will yield better results. So essentially what I'm doing is I'm isolating my feeders, I'm isolating my germ cells, and then I'm putting them together, waiting some time, and then kind of measuring my parameters for success. So in order to isolate my feeders, specifically for the myoid cells and the endothelial cells, I start with 1 week old porcine tissue. I do an enzymatic digestion that gives me a starting cell population. And then I use magnetic activated cell sorting, specifically for CD31 positive cells, which is for testicular endothelial cells. And that will allow me to separate my endothelial cells from my other cells, which consists mostly of myoid cells. And then I take advantage of differential adherence properties, and I do a differential plating step, which allows me to get rid of like Leydig cells and if there are any potential germ cells in that group. So I keep the adhered cells and I throw everything else away, and that should give me populations enriched for PMCs and TECs. And then I do a Sertoli cell isolation, which again, I start with 1 week old porcine tissue. And this time I do a different enzymatic digestion because the endothelial cells and the myoid cells are interstitial cells. So they're kind of in one part of the tissue, but these are within the tubules. So they require a different type of digestion. And then I'll have a starting cell population. Again, I take advantage of differential adherence properties and I keep the adhered cells and I get rid of suspended cells, which would consist largely of germ cells and Leydig cells. And then I have a population of cells enriched for Sertoli cells. And for the porcine fetal fibroblasts, I don't actually do that isolation by myself. I actually obtain those cells from 40-day-old porcine fetuses. I say that just because there's no kind of protocol that I've listed here. And before I move on to the next slide, I say that, again, I've done a lot here to enrich these cells, but that doesn't necessarily that mean that I've achieved the level of enrichment that I was hoping for these feeders. And I definitely found that I didn't. So I added additional steps that include passaging cells, or essentially keeping them in culture under cell type specific media such that I could increase the purity. So with the peritubular myoid cells, I had a very low purity at first, about 70% were myoid cells at the end. And then after my passaging, about 83%. The biggest jump was with the endothelial cells, jumping from just above 10% to above 95%. And then with the Sertoli cells from about 73% to above 85%. So now I have to isolate my germ cells once I've dealt with all of the feeders. And this time, instead of using 1 week old tissue, I use 8 week old tissue. And I do this because this is kind of the sweet spot where cells have, or the germ cells have made the transition from the progenitor to the stem cell, being gonocytes, to the stem cells, but haven't yet started to differentiate into less undifferentiated types of spermatogonia. So I start with my 8 week old tissue. With my largest potential population of stem cells. I do a 2-step enzymatic digestion that's similar to the Sertoli cell digestion. I have my starting cell population. I do a differential plating, except this time, instead of holding onto the adhered cells, germ cells actually tend to stay suspended in the media. So I keep the media, which gives me a population of cells enriched for germ cells. But there is still a little contamination. So I use fluorescence activated cell sorting. And at the end, I have a population of undifferentiated spermatogonia. And very quickly, before I move on, I just want to explain how we use fluorescence activated cell sorting, because we don't use it in the conventional way that it is often used. And like I said before, there's not a marker for spermatogonial stem cells. So how are we possibly able to use this? Basically, what we do is we have our cells, we bring it to the machine, and we kind of put it in this area here that has a probe. This probe picks up the cells and suspends it in fluid such that the cells are kind of separate from each other. There's just like a little bit of space that separates each cell from one another. And then they kind of go down this tube, and at the bottom of the tube, there's a laser. And this laser passes through each cell. And then on the other side, there is a detector. And this detector basically gives measurements of the cell that has passed through based on size and granularity, through kind of measuring the way that the light has scattered from the laser passing through the cell. And so what we found is that undifferentiated spermatogonia that contain SSCs will generally be very large and very agranular, or kind of empty. And so they tend to be in this kind of area here on the graph when all the points of each measurement are graphed. So what we can do is we can gate around this population, and then what the machine will do for us is give us an output of cells that only contains cells that would fall within this gated area. And this has been very successful for us. On the left, this is cells right after isolation. So again, about 7% germ cells, not all of which are stem cells. And then on the right, that number increases significantly to about 98%. And so once everything is kind of collected, it's time to put everything together so the real experiments can get started. So initially I was testing my seeding densities. So I started at 500,000 or 1 million germ cells per milliliter and 65,000 feeder cells per milliliter, except for the control that didn't have any feeders on it. And I measured the following parameters at weekly intervals. So I want to see how many of the cells are dying on each feeder, how many are dividing on each feeder, and if they are dividing on each feeder, how many of them are maintaining stem cell activity to ensure that I'm not just counting cells that are dividing, but differentiating. And I expect that the myoid cell or testicular endothelial cell feeder will be able to provide an environment for the germ cells to proliferate at least 15%, which is what we've observed in mice. That the cultured cells should express markers for germ cells and stem cells to show that they're not differentiating, and that they will, when transplanted, retain the ability to support spermatogenesis. Additionally, in comparison to the other feeder layers that I am kind of also investigating, I'm expecting that the PMCs or TECs will be at least as good, if not better, at allowing these cells, the germ cells, to grow and to survive. So some preliminary data that I have so far shows that the germ cells grown on the myoid cell or the Sertoli cell feeders have been able to proliferate significantly more in comparison to no feeders, not necessarily significantly more in comparison to the other feeders though. And not shown here was also some data that I was able to generate that showed that my two seeding densities didn't really make a difference on each feeder, or essentially that whether I plated 500,000 or 1 million germ cells on each feeder didn't really affect the way that the cells were proliferating. So going forward, I will only be keeping the 500,000 germ cell per milliliter condition. So to summarize this part of the presentation, porcine germ cell expansion will be really beneficial, and the results from this experiment or these experiments can be used towards expanding human spermatogonial stem cells. And this expansion clearly relies on me identifying the best possible cell type to act as a feeder layer. But it's also really important to understand why feeder layers support germ cell cultivation and why generally feeder layers are so beneficial in comparison to just growing cells in conditioned media alone. And that brings us to a very quick second part of this presentation, which is exploring the role of exosomes in co-culture conditions. So exosomes are a class of extracellular vesicle. Generally, they range in size from about 60 to 150 nanometers. For context, that is less than 1/1000th the size of the diameter of a strand of hair. And previously, they were thought of as junk because of their mode of biogenesis, and people didn't really think that anything could come of these vesicles. But recently, relatively recently, it was discovered that the cargo that's contained within these exosomes are functional, and that when these exosomes are exchanged between cells that are in close association, the cargo that's inside can actually affect the recipient cell. So I'm hypothesizing that the beneficial effects of growing cells with feeder layers are partially mediated by the intracellular communication that happens when feeder layers provide cargo through exosomal transfer to cultured cells. So to investigate this hypothesis, I'll be characterizing exosomes and comparing the differences between different feeders and then also the co-cultured cells. And I will not be getting into this protocol because it is, or these two protocols, because they are very long and a little bit boring. But essentially what I'm doing here is I'm starting with my culture media and then I'm spinning them at increasingly larger relative centrifugal forces, such that at the lower forces, I'll be able to get rid of larger things like cells and debris. And then as the force gets larger and larger, I'll be able to pellet things like the vesicles that I'm looking for. And then my characterization I'm looking to characterize not just identity, but quantity and contents. So to identify that I have exosomes. I'll be characterizing based on size and then exosome-specific markers. And then quantity and contents will also proceed with similar protocols. But I'm starting with my size characterization. And I did that using transmission electron microscopy. So a transmission electron microscope is a very, very large microscope, height-wise about floor to ceiling. And it's used to look at very, 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 very small things. So here I have an isolate from some peritubular myoid cell media. And again, 60 to 150 nanometers is generally the size of an exosome. And so this is the scale bar showing 100 nanometers. And so based on size, I can say that the things that I have, the vesicles that I've kind of noted with the red arrows are likely exosomes, but obviously further characterization has to proceed using exosome-specific markers. And I say markers because they're, again, similar to the spermatogonial stem cells. There's not one marker that can be used to identify all exosomes, but there are about 3 or 4 that can be used to identify about 99% of exosomes. So expected outcomes. I expect that the contents of the exosomes from my different feeders should be different. And something that I'm really interested in investigating in the future would be to assess the effects of these exosomes and their cargo on how germ cells proliferate. So I would expect that the cargo released from the feeders, as they are different or should be different, should affect germ cell survival and proliferation differently. And a final note that I'll leave you with is that I think establishing this system will be so important for human spermatogonial stem cell expansion, and also using these exosomes to really figure out what it is that's happening between the feeders and the co-cultured cells could also really benefit human spermatogonial stem cell expansion towards this final goal of being able to use these cells for transplantations to preserve the fertility of these little boys who survive cancer. Yeah, that's it. Thank you.
Thank you, Shiama, for that wonderful presentation. To our attendees, if you have any questions for Shiama, please use the Q&A box and we'll go through the questions at the end in our Q&A period. And now, Sumedh, you can go ahead whenever you're ready.
Sure. Can you see my screen?
Yes.
Okay, perfect. Good. Thank you very much, William, and thanks, Shiama. So hi, everyone. My name is Sumedh, and I'm a PhD candidate specializing in health services research in the Department of Community Health Sciences. So my presentation is going to be around KidsPRO, which is a tool to support patient and family-centered care in pediatrics. Yeah. So just to set the stage, when we talk about healthcare is usually refers to the organized provision of medical care to people and communities. Just like any other industries, in the area of healthcare or healthcare systems, it's important to have some outcomes. But what type of outcomes are important in healthcare? Are they clinical outcomes? Are they psychosocial outcomes? Are they financial outcomes? And why do they matter? Do they matter because of providing better clinical care, for improving quality of care, or for research purposes? And another very, very important thing is that if the outcomes matter, then from whose perspective? Because broadly, healthcare system has healthcare providers and the patients that are, and then there's an interaction between these two parties. So from whose perspective these outcomes matter? And if they matter, then how to assess them? So there is a tool, or there are type of tools that can be used to measure outcomes in healthcare, and they are called patient-reported outcomes. So the definition of patient-reported outcome is it is a report coming directly from patients about how they feel or function in relation to a health condition, and its therapy without interpretation, without interpretation by healthcare professionals or anyone else. So keep this in mind. What is patient-reported outcome? There are PROs. There are different types of PROs. So patient-reported outcome measures, PROMs, and patient-reported experience measure, PREMs, are the two major types. And I'll be talking about PROMs. So what you can see here is called PROM. So these are basically survey questionnaires, which are developed. They are standardized, validated questionnaires. They come in generic and disease-specific forms. What you can see here is a very commonly used PROM called PETSQL, so Pediatric Quality of Life Inventory. And it, as you can see, the questions on the right, those questions are organized in different domains. So around health and activities, around feelings, about getting along with others, about schooling. And the questions are very generic because this is a generic PROM. If it's a disease specific one, for example, for my PhD project, I'm investigating its use in asthma. So the questions are like, it's hard for me to breathe, or IVs, or questions around medications, like it's hard for me, or I don't want to carry my inhaler. So these kind of questionnaires are called PROMs. So they can be used for different things, but what my project tries to do is to investigate its use in actual clinical encounters, actual clinical care. But why should we use PROMs? Because PROMs, as I mentioned earlier, are questionnaires that place the value and perspective of the patient at the center. They capture patient's perspective. So it's a shift from the biomedical model of healthcare towards a person-centered model. And also it presents an opportunity to focus on what is relevant to patients to co-design their health. Now, this is based on also a framework that was proposed by my supervisor, Dr. Maria Santana, so this framework is to assess the effects of using PROMs in chronic care management. So completion of PROMs and sharing the results with clinicians and family members basically acts as a tool for better communication between clinicians, patient and clinicians, patients and relatives, clinicians and relatives, which leads to better patient engagement during the clinical encounters, which leads to better patient management, better decision making. It also increases clinician satisfaction. It also increases patient satisfaction and patient adherence to different type of treatments, and ultimately leading to better patient outcomes. So the use of PROMs is very common, I would say, in adult population, but its use in pediatrics is still lagging. So this was a paper that showed that why measuring patient-reported outcome measures in pediatrics has done a good progress, but there is still a long way to go. And there are several reasons for that. Some of the reasons are just this attitude among healthcare providers is, are children old enough to provide self-report? Can they, do they understand this kind of questionnaires, and can they complete this kind of questionnaires? So some research has been done in that area, and the minimum age at which children can self-report reliably whatever their health condition is considered age 8 years old. There is also an alternative where parent-reported outcomes are also used if they are very young. The level of cognition development versus PRO questions is another concern. So for example, for some conditions impact cognitive development of child, then how to assess PROM results from these children. Also in pediatrics, cognitive level is more important than chronological age, sorry. And also, so pediatric PROMs also come in different age-specific versions. So how to administer those PROMs in those specific age versions is another issue faced by healthcare providers. And so as children are young, the results are more similar, but between parents and children. But as children grow, as their personality develops, the results tend to diverge. If you understand PROMs, or if you get basic understanding of PROMs, then you'll understand what is KidsPRO. So KidsPRO or KidsPRO is an innovative healthcare information solution that supports and facilitates the integration of PROMs in routine clinical care for pediatric patients with chronic conditions. It is available to patients on mobile devices, tablets, desktop applications using a responsive web application software. And it enables patients and family caregivers and providers to work as a team to share their data before, during, and after the consultation. And it's been piloted currently at the Alberta Children's Hospital. To explain what KidsPRO is, we had created a small video. It's slightly over a minute, so I'll just play the video and we'll explain more about the project. So this is KidsPRO. I hope that gives you an idea what it is. We use it to recruit our patient participants and also to share, to inform about this project to others. When we started this project, one of the biggest thing that we observed was usually healthcare solutions or healthcare the different actors involved in this kind of projects usually work in their silos. So researchers would do their own thing. They might just take the findings to Alberta Health Services, or Alberta Health Services might just do their own thing and just implement there. It's not studied scientifically. Or industries would create some product, but they find it hard to get involved in the innovation process. And patients and family caregivers are not engaged as the members of the team who develop this kind of intervention. So we wanted to break these silos. So we partnered with all of them. And so this platform is developed by Cambian, which is a company, a private company that has developed the platform. Patient and family caregivers are involved. We are the researchers, and our study site is Alberta Health Services. So we are trying to break this silo. So first step we started with was patient partner recruitment. So these patient partners, we wanted to look for few people, 4 or 5 pairs of parent and child and their parents to act as the partners for our project. So they are our study team members, not the participants from whom we will collect the data. And this on the left is a picture you can see that's pre-COVID when we had this information cards at the Alberta Children's Hospital's asthma clinic to recruit patient partners. And we also created one more video to recruit patient partner, which is already on the website of University of Calgary and Alberta Health Services. I can control my asthma page, and that video is called Mission Possible. So I'll play that video next. The audio will take a few seconds. So as a result of these kind of strategies to recruit patient partners, we got a nice group of patient partners. So you can see in the picture, these are our patient partners, a group of 5 kids, youth, and their mothers. Our youngest patient partner is Nicholas, who you can see is in orange shorts. He was 8 years old at the time of this picture. And then Kim, who's the oldest one, she's now in university. She's at University of Calgary in her first year of a undergraduate program. So patient partners for KidsPRO helped us with recruitment strategies, with co-designing KidsPRO platform, with co-designing KidsPRO user guide, with co-designing KidsPRO logo. On the left bottom, you can see that logo. Provide feedback on systematic review. When we conducted a systematic review, their perspective was taken into account, added into the manuscript, appreciated by the reviewers, that patients' perspectives are taken into account. Patient recruitment and co-presentation there are 2 asterisks because of COVID. Some of these activities they were not able to engage, but we are going to do that next year. So first phase of the project was basically systematic review to basically synthesize the evidence to evaluate the impact of using PROMs in routine clinical care for pediatric patients with chronic conditions. And this involves all types of chronic conditions. The protocol of this review was published, and last year we published the findings of this systematic review as well. So, the results show that there are only 7 studies in the world from in last decade that that looked at that investigated use of PROMs and its impact on different outcomes in clinical care, like health-related quality of life or healthcare utilization or quality of care, patient outcomes. And what you see, NR is not reported. So there are very few studies that are studying this. And if they do, some of the results are very, very mixed. So that's where KidsPRO tries to generate some evidence around some of these outcomes. So phase 2 of the study is to identify barriers and enablers. So before the KidsPRO gets implemented in asthma clinic, we want to understand from the potential users of KidsPRO that what are the perceived barriers and enablers of integrating PROMs in routine pediatric clinical asthma care. It's a qualitative study guided by a theoretical domains framework. So approximately 15 healthcare providers are being interviewed. Actually, we finished interviews just now. Yesterday was the last interview. Study participants were pediatricians, pediatric respirologists, nurse educators, and administrators from Alberta Children's Hospital Asthma Clinic and from community asthma clinics. And this data is collected using semi-structured interviews, and they are transcribed. And currently, I'm analyzing the results. I have some primary results, but more interviews will be coded and analyzed soon. The strength from enablers will be harnessed, and the mitigation strategies will be devised using the data on barriers to PROMs. So these are the primary findings. So barriers are so far that we have been able to identify are lack of complete understanding to interpret PROM results. These are among healthcare providers. Time constraints and other competing tasks. So some healthcare providers mentioned that I don't have time to look at these survey questionnaires. I already asked these kind of things, but I didn't think this thing might take more time. Lack of integration within currently used EMR systems. So Alberta Health Services is rolling out a new EHR, actually electronic health record. So they are currently not integrated. That's another barrier. Enablers are actually healthcare providers who want to provide patient and family-centered care. So PROM, they understand the importance of PROM. They also want to understand the impact of asthma on patients' overall quality of life. So they know that PROMs would be able to do that. So this is the enabler. Also, systematic approach to understand patient needs and address those needs and monitor the progress over time is something that PROMs can help them to do. So these are some of the primary findings from the first stage of this, second stage of the study. Based on these results, we also have created user guides for patients and families to use KidsPRO system, and also user guide for healthcare providers. The logo that you can see here is actually co-designed by our patient partners. So we proposed few designs to them, and so one of them was even like emojis, and they said like, this is too childish, we want something like this, which is cool, and because you matter the most. So you are saying that the colors were chosen by our patient partners. So this is to show that how patient engagement can help in patient-oriented research. I just have two examples to share with you that how PROMs can help. And these are part of the user guides as well. So for example, a child, 13-year-old, has worsening asthma symptoms. So when she has to go to emergency for some condition, doctors ask, are you regularly taking your inhaler? She always say, yeah, yes, I do that. But when they completed the PROM before the appointment, she actually answered that I don't like to carry my inhaler. And she said almost always. So when parents, and healthcare providers probe further, they realize that, she feels shy to take out the inhaler in front of others or her classmates, peers. So she doesn't take it that frequently, which worsened her asthma. This is just an example. And this case is based on our patient partners' stories and their feedback. John, another case that we had added in our user guide is a 17-year-old teen who's socially impacted by asthma. So different perspective from parent and a child. So on the left, you can see the score in social aspect for this patient. Scored very low because he feels that he doesn't have enough friends. On the right is mother who completed the PROMs. She feels, no, my son has a lot of friends, and she doesn't understand the social impact of asthma on her child. So PROM results can also help you to, help you to better communicate between parents and patients as well. Phase 3 is currently underway, which is a feasibility assessment. So currently we're recruiting asthmatic children, 60 children and their family caregivers to try the pilot, try the KidsPRO and complete this PETSQL PROM. Healthcare providers are also being recruited, and all participants will complete the feasibility questionnaire after completing their completing these PROMs and using them during their clinical encounters. And different measures will be measured. So parameters, for example, uptake, compliance, perceived usefulness, experience using the KidsPRO and suggestions for improving. These kind of things will be asked to these participants. In conclusion, just a take-home message: PROMs are great tools to provide patient and family-centered care. KidsPRO is a platform to support integration of PROMs in pediatrics. Patient engagement is essential for patient-oriented research. As you can see, videos are very helpful for patient engagement and recruitment, especially in pediatric population. They might find other things very boring. And more results on feasibility of KidsPRO will be shared as the project moves forward. That's it from my end. Thank you.
Thank you so much, Sumedh, for your presentation. So at this time, I welcome any questions. You can enter them in the Q&A box. We have quite a diversity in presentations today, so should we, yeah. So we have a question here. Yes, this could be directed to both of you. So as fellow graduate students, have you had to adapt any of your methods to COVID-related shutdowns? And are there any specific experiments you haven't been able to do because of this and you wish to?
I can go, and then maybe Shiama can answer that as well. Yeah, for us, because our project is, we have to be physically present there. So we had to change our recruitment strategy. We had to change some of the protocol. We had to change the sample size, considering that I have to graduate. We don't know when will clinics open again. A lot of appointments are happening on Zoom. Earlier, a plan was to be present at the clinic with the tablets for patients to complete the questionnaire if they don't complete it before their appointment. So protocol-wise, a lot of things had to be changed. Yeah, and also presenting and disseminating the information. So a lot of conferences and local meetings have been impacted. So it's hard to disseminate the results. Luckily, we are working with the local collaborators within Alberta Health Services, so we are still able to convey our findings. But beyond that, it's very hard to do.
I guess for me, the two biggest things I think that have been affected are that, so obviously we work with primary cells, so we get tissue deliveries in order to be able to do our experiments. And, a lot of these isolation protocols are very long and time consuming. So in order to make sure there's not too many of us in the tissue culture room at the same time, we kind of have to take like weird shifts where some people come in very early and then some people end up staying very late. Also, right as I was starting to gear up to do more of my exosome work, the ultracentrifuge that I use broke. And, you know, in kind of normal circumstances, it would not be as difficult to get like a tech to come and, you know, figure everything out. But because of everything that's happened with COVID and all the limitations, it took months and months. So that was really stalled for a long time. Thankfully, you know, it's fixed now, but it definitely, like, I also feel like, you know, I go in more on the weekends now, or like we're just kind of trying to make sure we're not a big lab, but we do have to, you know, try and make sure we're not all in there at the same time. And that's been kind of difficult.
All right, thank you for the answers, Shiama. For Shiama, besides preserving male fertility, are there any other applications for these spermatogonial stem cells?
There are actually. So kind of along the same lines, everything that can be done with spermatogonial stem cells needs to be done through transplantation. But in addition to preserving male fertility, these cells can be kind of altered prior to transplantation. So this has been, again, only shown in animals, not for humans, at least anytime soon. But so they can be, for example, you can use CRISPR to alter the cell prior to transplantation. And because it's a germ cell or a sex cell, these changes that happen will be disseminated to future generations. And so these types of experiments can be used to develop models, animal models to study disease, and have been used actually quite often in, or are, you know, again, things are being developed, what's the word, like in farms and things like that, to ensure that, you know, certain types of livestock with certain features can be bred preferentially.
Thanks, Shiama. And one last question for Sumedh. How is patient engagement different in pediatrics compared to adults?
Yeah, so patient engagement in pediatrics, the way it differs from adult population is first the mindset of healthcare providers, that they have different mindsets. They think that to provide a patient-centered care for adult population is important to understand their perspective, but for pediatrics, their parents can make the decisions on children's behalf. So first is this mindset. Second is engagement of parents. So if a child is interested in providing feedback or becoming a patient partner, but if their parents are not, then it's a challenge. So parents' engagement is important too. Logistically, for example, for my patient partners, I always have to communicate to them, with them through the parents. So those kind of things are two important things that differentiate pediatric patient engagement and then adult patient engagement.
Thanks, Sumedh. So that's all the questions. That's all the time we have for questions today. Thank you to our presenters for sharing their research with us, and our guests for taking the time out of their day to join us and participating in the Q&A questions. Your questions were greatly appreciated. It was a pleasure to have everyone here join us for February Lunch and Learn. Feel free to connect with the Graduate College on social media and visit our website for more information about our upcoming events. We hope to see you all again soon. Thank you.
Thanks, William.
So we can go now.